speak. You have to speak in the mic because we're recording.
Hi, I'm Kathryn Homa. I'm also a graduate student, like Rob, at Rutgers in the Plant Biology and Pathology department. And I'm also a full-time fungicide coordinator at IR4 Project, which is also a part of Rutgers. I just finished up my work with Basil Downy mildew, testing fungicides and varieties. And so I figured it'd be a lot of fun to keep going and start on looking at different variety evaluations for control of Fusarium of Basil, which is Fusarium oxysporum basilicum. So Fusarium of Basil is caused by the pathogen Fusarium oxysporum basilicum, which is a fungus. It was first described back in Russia in 1957 and then later in Italy and France. It was not discovered in the U.S. until 1991 in Massachusetts, and most likely this contamination was thought to be from infected seed from Italy. Like Basil Downy mildew, it's less destructive, but it is a very common disease of basil that can be found in the field. It's greatly the most place it's found is soil-borne, but it can also be found airborne, and there are journals that confirm that. It can be introduced into the field or also through contaminated seed and also the movement of contaminated tools and soil. And unfortunately, this disease is most common in sweet basil. So the main symptoms, as everyone, growers have seen in the field, is wilting, and this occurs from the fungus attacking the water conducting system of the basil plant, which is the xylem. And once this is clogged, it results in several symptoms, most notably stunting and wilting. Also results in yellowing leaves, defoliation, discoloration of the stem tissue, and also a rot of the roots and the crown of the plant. So this is a picture of the common symptoms. As you can see, the stem is browning, it's stunted, and it's wilted compared to the healthy plant on the left. So as we heard before, New Jersey is a major grower of sweet basil, growing probably more than 600 acres per year, totaling $10,000 per acre wholesale, mostly in southern New Jersey, but also in central parts of New Jersey. We know that once this disease infects the basil plant, it dies, and very importantly here I want to note that the only chemicals for control are fludioxonil, which is seed treatment, and also soil fumigant, which, as you all know, soil fumigants are starting to go out of the market now. But 1,3-dichloropropene is still in the market. And most importantly, what we've just found out is that the resistant varieties may not be truly resistant. One of these examples is New Far, that's on the right here. So some of the other so-called resistant varieties, one is Aroma 1 and Aroma 2. These are the Genovese type cultivars, and a lot of them are used in production. It's important to note here that the camphor and lemon basils and others in the mint family, such as rosemary and thyme and many other important herbs that are grown, they do not display the symptoms, but they act as hosts. So this means the fungus can still be inside the plant and actually result in sweet basil actually obtaining the disease from these asymptomatic plants. It's also believed that Fusarium resistance may be a single dominant gene. It's important to look into that again. So the goals of the current study are to screen a number of experimental and commercial varieties for resistance, including varieties that are so-called tolerant or resistant to Fusarium. And if they prove to be tolerant or resistant, then they can be used in a breeding program to develop the new sweet basil varieties resistant to Fusarium basil and hopefully basil downy mildew, as Rob mentioned. So this was our Fusarium screening procedure. It was conducted at the New Jersey Agricultural Experiment Station Research Greenhouse in New Brunswick on Cook campus. We're testing 9 Fusarium basil lines for virulence. Big chunk of these Fusarium lines were obtained from the University of Massachusetts, so I want to thank Rob and Angela for those lines. We also got a few plants that were infected from Rarick and Pittstown, New Jersey at the Rutgers experimental stations, which are these 3 lines. And then also from a grower, Alan D. Young. I want to thank him for the line from Illinois. So far, it's been determined that almost every one of these lines is pathogenic. I'm still testing the one from Illinois since it took a while to grow out onto plates. So I want to go over the Fusarium screening procedure. First, we wash the PDA plates containing the growing Fusarium culture. We wash them with 10 mils of distilled water. And then using a microscope and a hemocytometer, which is here, we count the canidia using the hemocytometer and the Nikon microscope. So here you can see the squares, count each spore inside the square here to get a total. Then after we get the average, we dilute it to a 10 to the 6 concentration. Then we prepare the plants by washing the roots and cutting them halfway, drying them, and then dipping them in the solution for a minute and planting them. That's a picture of this procedure, which I hope to get faster results this way. It kind of takes a lot of time. We did try other techniques that we failed with before we had this other procedure that I was just talking about. We did, when we got the inoculum, we would inject the soil just in the pot with the plant with 10 mils. We would dip the root in inoculum for 30 seconds without wounding the roots. Then we went to wounding the stem with scissors and dipping for 30 seconds. And then last came the winners, cutting the roots and dipping for 1 minute, and cutting and wounding the stem and then dipping for a minute. Then we just went back down to just cutting the roots and dipping. The mistake we first made is that we tested mature plants such as this one first, and then when nothing was happening, they would stay healthy. We followed this by just inoculating at the 2 to 4 true leaf stage. So this is an example of the strain virulence. With 3 commercial varieties, we have Caesar, DeGenova, and Nufar. As you can see, with all of these lines, there were significant amounts of plant death observed after the inoculations, especially with the Rutgers lines, a few of the UMass lines, and that's another New Jersey line. So some resulted in 100% death. And as you can see, Caesar was the most susceptible, DeGenova was in the middle, and actually this is a big thing here, the one that we thought was tolerant, resistant, There were large quantities of these that were also had died from the procedure. So then, after the commercial lines, now we're starting with some of Rob's lines, different varieties. And a good thing here is that this one variety that was Rob mentioned was also with downy mildew resistance looked well. This also looked well for Fusarium resistance, where there was 0% mortality as the control and the treated. They both held up. And also with the plant heights to show that they weren't stunted. They have similar plant heights also. But with a lot of the other varieties that were tested, they showed 100% mortality, or pretty high mortality, compared to the untreated control. And then these are the different lines. We tested with 3 different lines to confirm this. Another variety that did well was the 652066. Compared to the control, there were 0% mortality, and the plant heights were pretty similar. And then they were also tested against these 3 Fusarium lines. With the cross that Rob was talking about earlier, where some of the plants survived and some didn't, that's the result of this cross. As you can see here, there was some mortality there compared to the control, so it doesn't show complete resistance. So I wanted to drive home this point that the commercial lines, Caesar, DeGenova, and Nufar, they are in fact more susceptible to Fusarium than we thought they were. The stunning and the wilting and death. These are the 2 lines that proved to hold up to the inoculation of Fusarium. They don't look too much like sweet basils, but they can be used for the breeding work. And also the picture that Rob showed, where there is possibly some resistance that we can get from this line here. So, like other presentations, I want to mention some other management practices, which is using treated seed. So there is that one fludioxonil treatment that can be used with Fusarium. Purchasing healthy transplants, good sanitation, throwing diseased plants away, increasing the spacing between plants with the air circulation, and also applying the irrigation to the base of the plants instead of overhead irrigation is a good idea if it can be done. Removing diseased leaves. And also an important fact here for, I know a lot of growers probably don't have enough of space to do this, but rotation might be a good idea to rotate with plants that are outside of the mint family, such as the rosemaries and thymes and mints for 2 to 3 years, and alternating that might also help with disease control. Oops. So that's my presentation, and if you have any questions for me or Rob, you can take them.
I was wondering about the data you showed for the different strains of Fusarium differentially affect several cultivars. Was that repeatable?
I didn't repeat it. I just got started, so we did this one. I did it with 3 different strains, as I showed in that table. So I think it would happen again because I did it 3 different times. So it was just with different Fusarium lines, and it all proved that in the same results pretty much.
Do you
field, it's spotty, so you can see that there's one wilted plant at the end of the row, and then there's not another wilted plant for several feet. So it's kind of weird how it occurs, but I think because it's a secondary pathogen, that plant has to have an initial wound in order for this pathogen to get into the plant. So it might have something to do with that individual plant being in the contaminated soil, and why not all plants are infected in one area.
Water is going to dictate a lot of that too in the soil.
A question for Rob. I think it answered the question by the time we got to the end, but I'm not sure. I was wondering whether or not you're confident that those unwanted essential oils are conferring the resistance to
Yeah, I think we can say that there's not a causal effect. It's possible, and this is one thing that we're going to do, is analyze those populations of all the oils to see if there's possible genetic linkage. But the fact that we've been able to get little rich, resistant sweet basils, would it be possible if there was a causal effect from, for example, methyl chaparral? But that's a good question, because methyl chaparral actually has been linked to antimicrobial function.
On your rating method where you said that one is 10% or less, is that all representative of the entire plant, the 6 leaves that picked because they look like they had?
Oh, that's a good question. Yeah, no, the leaves are picked uniformly from the first 6 leaves were picked uniformly, and the idea is that they do give representation of the entire plant.
And is there any knowledge whether the time you take the test is 10%, does that plant proceed to grow where it becomes 30%?
Yeah, right, so we do multiple ratings, and we pick the rating that has the highest quality.
So most of that time is 10%.
Yeah, so those ratings are made at the highest point. We don't use the area when it comes to progress curve. It's not appropriate for that. We take multiple ratings and then we look at the overall. Yeah, we don't do area under the disease progress curve, which is a function of taking multiple ratings and then doing a calculation based on all of those ratings. We want to look at what point are all of these genotypes, all these plants submitted to the highest disease pressure, and that's where you're going to get the genetic response of resistance. And that's also the conditions we make our selections under.
We could give everybody could give a round of applause to Kathryn for the Fusarium. Let me comment on 2 questions that Rob, you asked about the aromas. It does